Hi, everyone. My name is Brian, and I am a graduate student at the University of Washington. Today, I will present about my project titled Population Genetic Structure of Eelgrass Across Washington State. Zostera marina, commonly known as eelgrass, is a bed-forming seagrass widely distributed across the Salish Sea. These meadows form the foundation of estuarine habitats and provide several benefits to coastal areas and human livelihoods, such as shoreline protection, food web support, and nursery habitat. Because of their recognized importance, a tremendous amount of management effort is dedicated to eelgrass conservation, including large-scale transplantations of plants to mitigate local declines. However, these practices are hampered by a lack of knowledge about the population structure of local eelgrass. In other words, plants are all treated equally, without addressing whether their genetic diversity is unique or redundant, which populations best serve as transplant sources, or how would such transplants influence current structure and diversity. In order to develop successful conservation strategies, a key step is to determine baseline population genetic structure relevant for the designation of management units. To fill in this critical knowledge gap, my research aims to describe the population genetic structure of native eelgrass populations in the state of Washington. Through this work, I will be able to generate a detailed map of genetic variation across the state, and this, in turn, will enable us to inform management by identifying populations with unique genetic diversity to protect or suitable donor sites to propagate. For this work, we sampled 16 populations, 14 of which are in the Salish Sea and 2 on the Washington coast. These sites represent 4 different oceanographic areas where eelgrass is found: the northern and southern basins of Puget Sound, the Hood Canal, and the Pacific coast. We used next-generation sequencing, or RADseq if you're interested, and genotyped 214 variable DNA site genetic markers, or SNPs, that is, variations on single nucleotides in the DNA. Our initial dataset consisted of 753 individuals, but only 436 remained after filtering out clones. This is a common practice in the study of asexually reproducing species to prevent confounding results in the study of genetic diversity. With this data, we described shared genetic variation by conducting a discriminant analysis of principal components that clusters individuals based on genetic similarity. In this plot, each dot represents an eelgrass sample colored by population. And the closer 2 points are to each other in the graph space, the more genetic variation they share, or in other words, they are more genetically similar. In this graph, we observe 4 major clusters of samples. The most evident grouping is formed by the Washington coast populations in blue. In Puget Sound, the northern basin, circled in red, and the southern basin, circled in orange, show evidence of divergence. And finally, geographically distant eelgrass populations in the Hood Canal, circled in purple, are also genetically isolated from the rest of Puget Sound. Here, the x-axis that explains the most variations within the data separates the coastal and Puget Sound populations to opposite ends. The order of the populations on the y-axis instead only partially conforms to a north to south direction for the Puget Sound clusters. We found extensive genetic structuring at the regional level that reflects geographic spatial patterns. These findings suggest high connectivity among meadows within areas, but not between them, especially between North and South Puget Sound, but also between the Hood Canal populations and the rest of the surrounding water body. This type of structuring in the same watershed could be caused by barriers to gene flow, such as currents, or maybe the distance is just too long for successful seed dispersal to occur. We also observed that eelgrass in the Salish Sea is genetically divergent from the Washington coast, and all this information is highly valuable to designate appropriate management units across the state that will support science-based decision-making. We also found a range of clonality rates from 0 to 900% of sample individuals being clones within populations. 4 sites were dominated by clonal reproduction. However, we are still analyzing how these surprisingly high rates of clonality might affect the standing population structure and what their contribution is to overall metabolic stability. Thank you.